Welcome back for more anatomy and physiology. We're going to deal with blood vessels today, and in particular, how do structure deal with function with regard to blood vessels. So as we make blood flow around the body, we can control, or we can describe this using Poissier's law. Poissier's law deals with flow through pipes, which is pretty good for circulation. So what we get is a relationship between flow, Q, and pressure and the diameter of the tube and how long the tube is and the viscosity of the fluid in the tube. The point of this is whatever happens to the tube or the contents of the tube, the heart has to make it up. So it matters how the tubes are built. When I look at the blood vessels, there are 6 of them. 5 of them actually are blood vessels, 1 of them is not. What we start to notice is that they have a similar pattern in how they're built. And they're composed of 3 layers that we call tunics. We have the tunica interna, which is the inside layer. It's made out of simple squamous epithelium. We have the tunica externa, which is the outside layer. The tunica externa happens to have a lot of connective tissue associated with it, so it can connect to all sorts of things and hold the artery or the vein or whatever in place. Then we have the tunica media. The tunica media is primarily smooth muscle for that middle layer, and there might be other things associated with it. The difference between the artery and the vein is how we build those 3 layers. If I strip away the outer 2 layers and we just leave the tunica interna, what we get are capillaries, or in some cases lymph vessels. Looking at an artery, what we notice is their function is to maintain pressure. The way that they do that is they happen to have smooth muscle, which will shrink down in size, but they also turn out to have elastic tissue inside of them. And the job of that elastic tissue is when it gets stretched, it wants to go back to its original shape, because that's what elasticity is. If you distort it, it wants to return to its original shape. How are we going to do, or what does this mean? You have a tube, an artery, blood gets squirted into it from the heart, it swells up, and then the elastic tissue brings it back down to its original shape. It's not going to be an immediate snap, but it's going to be a slow, steady push. The result of that is we're going to eventually have, or will maintain, blood pressure. Arterioles are small arteries, and I know that because arteriole means little artery. What we notice with these is they are built very similar to arteries. The exception is they have an extra ring of smooth muscle on the outside. The result of that is that smooth muscle ring can contract. The result of that is we can change the diameter of the vessel. If I can change the diameter of the vessel, I can control blood flow because of Poissier's law. What do arterioles do? They control blood flow through capillaries. And we'll talk about how they do that magic trick later. When we look at capillaries, they're composed of only simple squamous epithelium. You have more capillaries in your body than we can possibly begin to describe, to the point where any cell in your body, it is about 2 cells away, no more than 2 cells away from a capillary. Meaning I can have capillaries and I can stick at most 2 cells in between the capillaries. That's how many you have. How big are cells? They're dinky small, which means we have a lot of capillaries all over the place. Capillaries turn out to be the only place where exchange can occur from the tissues to the blood. So if I want to put something in the blood, it can only happen at a capillary. If I want to take stuff out of the bloodstream, that can only happen at a capillary. That is the location. Why? Because one of the jobs of simple squamous epithelium is it's a filter. It's just there as a barrier. What we notice at capillaries is there's an artery side and there's a venous side, a vein side. And what happens is we tend to have fluid exchange. Stuff tends to move from the bloodstream out on the arterial side, and fluid tends to move from the outside world, the tissue side, back into the bloodstream on the venous side. Turns out it's not entirely efficient, but we'll get to that in a bit. When we look at what comes next, venules and veins, venule, tiny vein, they're built the same way. They turn out to have a lot of smooth muscle because smooth muscle is capable of stretching, but it doesn't necessarily need to go back to its original shape. The result is These are good for holding on to fluid. So if you look at this picture, what it shows us is the veins hold about 64% of your blood at most times. Capillaries about 7%, arteries about 13%, pulmonary side is about 9%. Most of your blood sits in veins, and because they're large and baggy, that's perfectly okay. To help them out, we have to add valves, because those valves ensure that blood moves back towards your heart, and we want to make sure that blood's always going forwards and not backwards, and the job of the valve is don't flow backwards. I told you earlier at the capillaries that fluid dumps out on the arterial side, comes back in on the venous side, but it's not a perfect system. It turns out we're missing fractions of a percent in terms of the exchange, meaning not all that dumps out comes back in. That doesn't sound like it's a big deal, except that within a day it'll kill you. You'll actually run out of blood. You'll excrete away enough water that we're going to have issues with blood flow. So what we happen to have is an additional vessel referred to as a lymphatic vessel or a lymph vessel, and its job is to pull in that remaining liquid. It also does a few other things, but for our purposes right now, its job is to reabsorb liquid. When we look around the body, there's lots of arteries, there's lots of veins. They all happen to have names. So what we're going to start to work on in class is how can we name these things, and can I use the names to connect body parts? So that's where we're going to be dealing with in class next.